I'm John Cook, Chair of the Department of Cardiovascular Sciences, and I'm here with DEEPAK Srivastava, who is a distinguished professor at UCSF and also the director of the Gladstone Institute for Cardiovascular Medicine. And he just gave a brilliant talk to us today on cardiovascular fate, the determinants of cardiovascular fate, and how that understanding has led to understanding of the mechanisms of disease, aortic valve disease, septal disease, and also may lead to new therapeutic avenues for treating patients after myocardial infarction. You're going to hear about that in just a minute from DEEPAK. But I'd like to start by asking DEEPAK, he's been so successful in cardiovascular medicine and biology. What were the seminal events in his life? Who were the people that directed him down this path? Tell us a little bit about that. How did you get to where you are now?
Well, that's a good question, and actually, oddly enough, I can point to very discrete days in my life where there are pivotal decisions, starting as early as college, where I was here at Rice University across the street and was an economics major and thought I was going to go to Wall Street and be an investment banker. And one late night, a girl, it's always a girl, kept asking me questions about why I was doing what I was doing, and by 4 in the morning it was crystal clear that I should go into medicine, and not just medicine, but do science and medicine to try to help people. And so I changed all my classes the next day, literally, halfway through college, and ended up going to medical school.
Oh, spectacular.
And I guess...
and that was at UTMB, Galveston.
This was at Rice University, and then I went to medical school at UTMB in Galveston. And there, I was greatly influenced by a research mentor, Brad Thompson, who was also a Rice grad decades earlier. And I worked for about a year in his lab during medical school. And on my graduation, for a graduation gift, he was a sailor and he had taught me how to sail. He took me out on his boat. We did an overnight trip, just he and I. And I remember, again, late night, I asked him a question, I was probably a bit of a, cocky 24-year-old at the time, and I said, if you were a hotshot, young scientist right now, what is the big thing out there that you could pursue? And this was 1990. And he thought about it for a while, and he said, I think I would try to figure out how it is that you start from an early embryo where all the cells have the exact same DNA, yet distinguish themselves into such different types. How is that controlled transcriptionally? And he studied steroid hormone receptors, so nuclear regulation. How do those cells know to become what they're going to become? Yeah. Which, if you think about it, is what I still study to this day. So I think the scientific question got implanted very early at that time, and that's something I've been passionate about ever since.
And so you went from there to some great places. You were at Children's Hospital in Boston. You were at UCSF, got some training there as well. I ended up as a professor at UT Southwestern. And you've continued to follow this track of trying to understand what are the determinants of cell fate. You gave us a great talk on that. Tell us, for those people that are watching right now, they can certainly go look at your talk, but there are some things that you didn't mention in the talk that might be worth a little bit more discussion. Tell us a little bit more about your initial work with microRNA and how that led you to understanding its role in cardiomyocyte fate.
Sure. So as I went along in my training as a clinician, what drove a lot of our discovery in cell fate and the genetics of it was my clinical experience taking care of children who were born with heart malformations, and parents, saying, "Doctor, why did this happen?" And we had very few answers. And so over time, that led us to figure out, how the heart forms, then trying to understand if those genes are mutated in the setting of disease. And repeatedly, we were finding that those mutations in humans that cause disease were just decreasing the dose of those proteins by a little bit. And so what became obvious then is that the precise amount of proteins that are being made from these key genes was critical. So then you ask then, how is that regulated by the cell? There must be some exquisite regulation of the protein dosage. And it's that observation actually that led us in the early 2000s, just as the microRNA field was emerging, and within maybe a year of it being even described to be present in humans, which was 2001, we began to ask, are there any microRNAs in the heart that would, in some specific ways, could be influencing formation of the heart and the dosage of these key pathways. And so that's what led us down that road initially. It was the genetics that drove that question. And then over the subsequent years, we identified a number of cell type enriched microRNAs for cardiomyocytes, endothelial cells, smooth muscle cells. And in each case, those microRNAs turned out to be embedded in the major transcriptional networks that are controlling that cell, either its fate or its response to stresses. And so I think the overall overarching theme that we found, and others have found this also in other tissues, is that the microRNAs are essentially reinforcing the major decisions that the transcription factors are trying to make the cell do. And in various positive and negative feedback loops, they're reinforcing that decision in a critical fashion.
So I think this understanding that you helped us to achieve is going to be really important for cardiovascular regeneration. At the moment, there's a lot of excitement about cardiovascular stem cell therapies, and the problem, though, with that approach is, well, there's a lot of problems. One is delivery and injecting cells into an ischemic zone. For example, in the case of someone who's had a heart attack, a myocardial infarction, the idea is to try to, with cardiovascular regenerative therapies, try to modify the fibrosis, the scarring that occurs. And to achieve that, injecting cells it's a bit difficult proposition, and for a lot of reasons, which we won't go into. But I quite like the idea, your idea of modifying the cells that are already there. Could we change the fibroblasts, for example, into cardiomyocytes or endothelial cells, improve the perfusion, reconstruct the heart from the cells that are already existing there? And fibroblasts, certainly, there's a lot of them. They produce a lot of scar tissue. Is it possible that we can transdifferentiate those fibroblasts into something else and get true tissue regeneration? Talk a little bit about that.
Yeah, the observation you made is one that we've thought a lot about. Our lab and many others have been trying to transplant cells into the heart for various regenerative approaches. And I think some organs in the body are going to be more amenable than others to cellular transplantation for regenerative medicine. The heart poses unique challenges because of the contractions it's undergoing and the integration that's necessary for the cells that you introduce with the existing tissue to make a difference. And so, because of those observations, some years ago we turned to this alternative approach to harness the cells that are already in the organ and make them do something they don't normally do. And essentially, we're trying to control their fate to our advantage by hacking the code, if you will, of the cell and altering making them turn into something they wouldn't normally be. So we've had some success in doing that, and I think there are further improvements that can be made in that process, but I think it's been sufficient to begin to think about how to now refine that as a therapeutic product that it could enter a clinical trial, given some large animal data we have that looks promising. And so we have been able to license that technology to a startup company and obtain commercial investment to begin to do the translational work that's necessary to file an IND and get started on a clinical trial. So I think there's a lot of potential there, but it also has its challenges of delivery and safety that have to be monitored carefully.
Let's talk a little bit more about that, because the Houston Methodist Hospital and the Research Institute are very translational. We have a research institute that's integrated with the hospital, and we have a mission to perform fundamental research, but also to translate that research into something that's useful. So we have a GMP facility up on the 12th floor of the Research Institute. We have programs toward developing products. I think we'd like to hear more from you about what is the role of scientists in development of the intellectual property and its commercialization. Tell us what your thoughts are about that process.
Yeah, it's a very important point, particularly as physician scientists. In our group and at the Gladstone Institutes, we do basic research, but very related to human disease. And our mission is to identify the fundamentals of human disease in order to help mankind. And I think some years ago, it was sufficient for us in that academic setting to make great discoveries, find new targets of disease, and somebody would come along and pick that up and take it to the next step, and we would continue in our space. I think that all fundamentally shifted after the 2008 recession when capital became more expensive and the commercial sector became more risk averse. And I think we have certainly observed that in the absence of greater proactive measures, we could keep making great discoveries and they would just sit on the shelf. And then we would not be fully fulfilling our mission. And so we at Gladstone have actually created some structure around this to address it. We don't do the translational work itself at our place, but we have hired several business development experts from the industry to help us with our intellectual property. We've tried to be creative about structures that we could de-risk the landscape for commercial investors either in the form of startup companies, like what we've done for the cardiac regeneration work, where we have several founders from Gladstone that are going to deeply be involved in helping a company take it to the next step and de-risk it that way, to de-risking by creating partnerships with biotech so that we continue to provide the expertise, but partner with the types of expertise they could bring on, whether it's drug discovery or other areas. And even partnering with pharma in creative ways to take our discoveries to the next step. So I think the time has passed where we can just be bystanders with our discoveries if we really want it to make a difference for people. And so we're very active about that. And I think the role of the scientist is shifting so that it shouldn't be, it's not for everybody, but for those that are particularly passionate about their discoveries potentially being translated. I think in academia we have to be creative about how we enable that.
That is that's wonderful, wonderful news, and very exciting to see that your work is moving toward the clinic. Thanks so much for joining us today, DEEPAK.
Thank you very much.